Hello, I'm Dr. Frances Colles from the University of Oxford. Today I'm going to be talking about a brief history of Campylobacter research using the PubMLST database. So much of the work that I'm going to be presenting today is that done by Alison Cody, or Ali as she was known to many of us. You'll see that Ali's contributed a huge amount of work towards genomic research for Campylobacter. She's also spent many hours curating the database, and I very much miss being able to go and ask her questions about curation and the typing schemes. She's also been really kind to me over the years, helping me to get to grips with writing up my PhD, learning to make presentations, and we've also travelled to several conferences together. So you'll see from the picture outside of work, she was a keen horse rider and she loved nothing more than going for treks in the Welsh mountains. And as you can see, she rode in any weathers, so not for the faint-hearted. So I'd like to dedicate this talk to Ali and her family. So I'm sure most of you in the audience will know that Campylobacter is the most common bacterial cause of human gastroenteritis worldwide. Historically, there's been some problems trying to trace the source of infection because we were reliant on techniques such as serotyping. So Campylobacter can change its serotype and gained a bit of a reputation for being genetically unstable. So together with Kate Dingle, pictured here, and Martin Maiden in 2001, we published the first multi-locus sequence typing scheme for Campylobacter jejuni. So this is a DNA-based sequence typing scheme, which is more reliable. We sequence short sections of DNA from 7 housekeeping genes, which combine to give us a sequence type, and this is how we distinguish between different strains of Campylobacter. So we quickly realized from the large isolate collections, when we're trying to trace sources of human infection, that there were clusters of closely related sequence types, which we've called clonal complexes, and several of these were associated with particular host sources. So for example, ST61 and 48 complexes shown here are predominant amongst ruminant sources. 257 shown here, predominant amongst chicken source, shown in light blue, with a couple of environmentally associated clonal complexes. And then there's 2 large clonal complexes, ST21 and 45, which have a more generalist lifestyle and infect a wide range of host sources. So unfortunately for humans, shown in red there, we seem to be able to be infected by any type of Campylobacter from any source. Generally, we'd see less from environmental sources, which probably is a reflection of how commonly we're exposed to these. So we know from these types of studies that most of human disease is coming from contaminated chicken meat. So then the question is, where is chickens getting their Campylobacter from? So the perceived wisdom is that it's coming from the environment, with wild birds being a potential source. So we tested on the university farm for part of my PhD the Campylobacter populations from free-range chickens, which are very definitely interacting with their environment, and then these 2 species of wild birds, which are present on the farm in very large numbers. So we needed to test large numbers of samples because we know that Campylobacter is very genetically diverse. So for example, we collected over 1,000 samples from starlings in the middle there. And then we found, using the 7-locus MLST typing scheme, that the Campylobacter populations are very distinct between these different host sources shown in the graph there. So actually, when we did some more work with Jonas Waldenstrom, Petra Grieksborg, and also Nigel French, we found that actually wild birds, even from different countries or continents, would be more likely to have the same Campylobacter genotypes than these different bird species on the same farm. So Campylobacter jejuni has a number of different clonal complexes. We can see it's a little different in C. coli. Here we have C. jejuni on the top of this tree shown in grey and C. coli at the bottom. So they generally share about 80% nucleotide sequence similarity. And then we see that C. coli generally splits into 3 large clades, shown here in blue, yellow, and red. And then sometimes when we're looking at the sequence data, we'll see a mix of alleles from jejuni and coli. For example, with ST61, the 3rd one down there, 6 of the alleles are derived from C. jejuni, and the last one, UNC17, is from C. coli. So this isn't a mixed isolate, it's legit. And then sometimes with this C. coli isolate at the bottom here, we'll see mosaic alleles. So they've derived pieces of sequence from C. jejuni and then other bits from C. coli, as you can see in the sequence alignment here. Sam Shepherd's done some more work on this, so it looks as though there's a role for agriculture, intensive agriculture, sort of driving the evolution of Campylobacter species. So he's going to be talking a bit more about this later on. So we have a unique collection of Campylobacter human disease isolates in Oxford, spanning 18 years now. So it started in 2003 by Kate Dingle, and then Ali's literally collected thousands and thousands of isolates over the years. And more recently, Sophie and I have continued the collection. So these are human disease isolates that come through the door of the John Radcliffe Clinical Microbiology Lab. And you can see that despite efforts to reduce the huge numbers of infections that are out there, we see very little impact. There's possibly a bit of a golden era between 2010 and 15, but since then, the reporting procedures have changed for Campylobacter, and also the identification, which is currently done by PCR, and then the lab send us the isolates from positive samples. And you can see even the COVID pandemic hasn't dented the numbers of Campylobacters that we're still seeing. Sometimes alongside the Oxford collection, we do other studies from matched populations. So we've looked at samples from Newcastle in the northeast, Nottingham and Hampshire. So this has been really helpful to establish that what we're doing in Oxford is applicable on a national scale. So Ali was instrumental in setting up the core genome MLST scheme. So this is similar to the 7-locus MLST scheme, but like on steroids. It allows us to do some very fine typing. And we now have 1,343 genes included in this scheme. So these are genes that are shared by 95% of C. jejuni and C. coli isolates. So I'll demonstrate in a minute what we were doing with this, but this is a screenshot from the PubMLST database showing just a small number of the loci and alleles there. So in some of our earlier work, we noticed that some of the sequence types were stable over a number of months or years even, and so we wanted to investigate this a bit further. So we were able to do this using the core genome MLST scheme. So during 1 year's collection of isolates, we compared those from Newcastle and Oxford, and we were found closely related cluster between the both. So they were only different at up to 25 alleles, so that's less than 2% of the core genome alleles. And then when we compared this with other isolates on the Campylobacter database, this time spanning 8 years, the distance only increased to up to 30 differences. So we're seeing some very stable, very closely related single linkage clusters in the food chain. And the fact that we're picking them up in the northeast and Oxford probably reflects the sort of national distribution of food. Ali, this is one of the last publications that Ali did. So she did a systematic review of source attribution of human campylobacteriosis. And using several different models, they predict that between 44 and 83% of human disease is derived from contaminated chicken meat. And then the next most common source would be ruminants. So Noel's going to talk a bit more about this later on. Ali's also published some work looking at the potential of human disease isolates to come from wild bird sources. So I need to emphasize that these graphs are starting at 94%. So over a 10-year period, we see a very small but consistent amount of human disease isolates that can be attributed to wild birds. So between 2 and 3.5%, which sounds small, but because of the very large number of isolates and human disease out there, that's equivalent to about 10,000 cases nationally a year. And we do see some seasonal increase in the summer, which again probably reflects people spending more time in their gardens and more likely coming into contact with wild bird feces. But this isn't enough of a difference to drive what is known as the seasonal peak in Campylobacter. And then finally, in the human disease collections, we're seeing rather an alarming increase in antibiotic resistance. So in the most recent studies, we've kind of gone back and looked at some more historical isolates as well as more recent isolates. And these are antibiotic resistance profiles predicted from the whole genome sequence. So we're seeing an increase with both fluoroquinolone and tetracycline resistance over the years amongst Campylobacter. And then in a UK-based retail meats survey, we also see high levels of fluoroquinolone resistance amongst chicken sources, and also tetracycline resistance, and also tetracycline resistance amongst ducks and pigs meat. So to summarise, Campylobacter jejuni clonal complexes are largely host associated, whereas C. coli forms 3 large clades. They're large clusters of genetically stable Campylobacter in the food chain that can persist over many years. Most human disease is derived from contaminated chicken meats, though other sources of infection can't be ignored, and the levels of antibiotic resistance are continuing to increase. So I've very, very quickly whizzed through 20 years worth of research. We're very grateful for the organisations listed at the bottom there for funding us during this time, and also for all of the number of people that we've worked with. Thank you for listening.